to get started, thank you everyone that's present today on our 8th lab talk, a dive into flow cytometry, developments and applications to scientific research. My name is Sam, and my background personally is in biochemistry, and I'm currently a lab specialist at Cluster Market. We are the leading equipment scheduling and management solution for R&D labs, and today I will be moderating this talk. Thankfully, we have two wonderful guests with us that you heard chatting before we started, and I will do a quick introduction of them. We have Christiane Hassel. She's a flow cytometry core facility manager at Indiana University, and her background is in biology. She has completed her master's of zoology in the lab of Dr. Brian Calvi, studying induced endoreduplication in Drosophila. Her first introduction to flow cytometry was around the early 2000s with a fax caliber instrument that also sorted. Christine has been managing the IU Bloomington flow cytometry core facility for almost 16 years now, and they work with a variety of cells and organisms, including microbes, archaea, C. elegans, Drosophila, snails, human, mouse, and other mammalian cells. The facility has both conventional flow cytometers and sorters, in addition to a large particle sorter, which is the Copa Select. This can sort whole organisms such as C. elegans at all stages and Drosophila embryos, in addition to pollen samples. So I think she brings a lot of different sorting expertise to the table today. We also have Raif Yuecel. He's the head of the Center for Cytomics at the University of Exeter in the UK. Dr. Raif has over 20 years of experience in the field of cytometry, both academic and industrial levels. He has completed his PhD and postdoc work at the University of Duisburg-Essen, where he gained his passion for cytometry technologies. He worked at the University of Aberdeen as the head of the Ian Fraser Cytometry Center. And for the industry, he was the cytometry study director at Philip Morris International, and also the product manager for in vivo imaging and cytometry at Milten. In 2019, he joined the University of Exeter as the cytometry lead PI and head of the newly founded Center for Cytomics. The Exeter Center for Cytomics is fully operating since March 2020, and it has state-of-the-art cytometry technologies, including the Attune acoustic cytometer, the Aurora spectral cytometer, Amnis imaging cytometer, ImageStream X, MK2, and Luminex system FlexMap 3D. For sorting of cells and particles, the center offers BSL-2 level sorter fax area fusion and the latest state-of-the-art BSL-2 specter sorter Bigfoot. So I welcome both of you with your tons of years of experience, and where I'm very happy to have you both here to chat about flow cytometry and for everyone else to tune in. Just to get started, what are some of the new technologies that are being developed right now and that are up and coming within the field? Could you share something, Raif?
Yeah, sure. Thanks very much, Sam, and the entire Cluster Market team actually to organizing these webinars and giving me the opportunity to discuss and chat actually with Christiane. It's a great pleasure to be with you in this panel, Christiane. So it's nice to chat about the cytometry and this entire platform.
Yeah, thank you, and thank you as well for to Cluster Market for the invitation and being able to for the invitation here and being able to do this. And yeah, I think it's gonna be a fun conversation, Raif. So looking forward to it.
Absolutely. So it's really nice to be here and giving the opportunity about this. Actually, in general, flow cytometry is the technology, the cutting edge technology. I mean, I'm sure Christiane will confirm this as well. The single cell technology in general, all this technology or instrumentations are already quite sophisticated and really getting into the next levels of this multicolor assays or multicolor polychromatic flow cytometry. But recently, I mean, at the beginning of this flow cytometry, it was more about the hardwares to get more lasers, to get more dyes, and then to get more analyzing subtypes of immunological cells. And of course, it's not only immunology, but also, that's really great. We are sharing the similar application fields like we are also very heavily involved in marine microbiology and botanical cytometry. And I'm glad that I'm sitting here with an expert in this field as well. So, but recently, over the last years, the spectral flow cytometry actually is really making a big step forward from the hardware perspective, because it allows now us to measure the entire spectral signature fingerprint of the individual dyes. And this allows us not only from the immunological perspective, not only 20 or 30 subsets, but it goes now beyond 40 subsets of immunocells. And of course, this is on the hardware side, but at the same time, we need to consider the software side. That's,
Yeah.
The software is another topic. And I mean, it's not only the technology on the hardware, it's the software. I mean, what do you think, Christiane?
Yeah, so I agree. So spectral allows for that increase in number of dyes, expansion of panels, but as you said, at the same time we need the software and also having the software that can learn and understand and be able to give us information and without user bias.
Oh, yeah.
Right. So that's super important as well. So those technologies that are being developed to do that data analysis, I completely agree. And something that I would also add, besides the spectral, because now we have, like you said, we not only have spectral analyzers, but we now have spectral sorters. So that now increases our capabilities as well for increasing panel and the design, but the design is also now super important. So we're going from the initial, we have to design the panel, make sure it works, then put it on the instrument, whether it's conventional or spectral, and then sort it successfully, or if we sort it or analyze it successfully, or both, or we have both. Another technology that really came up in the earlier this year and was really prevalent at the Cyto meeting was the imaging sorter. So where you can look at your sample, it shows you the image of your cells or particles, and then you can sort based on that. And I don't know about you, but ever since they came out with an image stream, however many years ago it's been now, that AMNIS came out with the image stream, I don't know about you, but I have been wanting something that can sort based on imaging since that time. And we finally have something from BD that has put that out. So I'm really curious to see how we go forward with that, because and how that will be used and what advantages that will give us as well, because that adds another dimension to being able to analyze a sample and not only see it based on scatter and fluorescence, but also see it based on morphology and being able to see internalization, localization, things like that. And so that's just another step forward. And I'm super excited about all of these things that are happening.
So just to recap, so we have some news on the hardware side, right? The spectral and the imaging sorters, and on the other side, we have advancements in the software and the data analytics capabilities. Just as a follow-up question of the imaging sorter equipment, what do you think will be the impact for microscopy, for example? Do you think this in a future will be able to replace microscopy in general and do both at the same time?
Yep. You wanna go ahead, Christiane?
Yeah. I think I think it won't ever get down to that resolution. So we're not looking at a confocal microscope. We're not getting that high resolution. It's going to help, but it's not going to take the place of microscopy. It's going to, I think, help our flow cytometrists and in, again, parsing out some more information about the cells or particles that we want to sort, to give us more information. But I don't think it'll ever replace microscopy. It will be another complement to it, but it won't get down to the resolution. Well, I can't say that it would never get down to the resolution, but for the speed that we would want for the analysis and the sorting, that won't. I don't foresee it replacing microscopy at all.
I fully agree with you, Christiane, and I'm sometimes surprised about the resolution of the image stream. To be honest, I mean, it's interesting, but the advantage in the flow cytometry, I mean, as Christiane said, the resolution we will never achieve that, because for that to achieve that, you need a really super camera systems, and then also slowing down. You can't, and flow cytometry is I mean, the predominant advantage of flow cytometry is on a single cell level in a high speed way. So the image stream is going up to 5,000 or 10,000 cells per second. It's a compromise between the microscopy and the flow cytometry. But the advantage of I mean, in a flow cytometry imaging is the quantification, the numbers that we want, and the amount, the high throughput, the numbers and the features and the parameters that we want in a flow cytometry world. And it's not about getting the images, it's more about what can I get from the images. And in the traditional flow cytometry, you get only the fluorescence intensity or the scatter intensity. And if you look into the microscopic features from image stream, you get huge amount of quantitative data, and that's quite powerful. I don't think it's true that we will get to the question is, do we need that? So this is another. Do we need really this high resolution in a flow cytometry? I don't think so. So I think it's a kind nice symbiotic between microscopy and the cytometry world, flow cytometry world, I must say, because I believe that the microscopy belongs to the imaging cytometry world. So in the flow cytometry. But what will happen, and that's really interesting, it's the the increase of the image cytometry technology, like multiplexing image cytometry technology, like the one from Miltini Maxima or 10x, to get more parameters on a spatial resolution level. Still not the resolution as a confocal, but having more than 40, 50, hundreds of markers on a tissue section and analyzing them on the quantitative level, like in a flow cytometry, using really high dimensional data analysis tool for that. Like, I mean, there's a lot of on the market, not only Miltini, Akoya or 10x also started this with analyzing tissue samples on a nucleate and protein level. So I think this is it's going to be, the spectral made a big jump, really a big bump in the flow cytometry world. That's really, but the other on the hardware, probably the hybrid technologies getting more like you know, omics world, the imaging flow cytometers, or here, you know, the Hyperion, you know, imaging mass spec flow cytometry. It's a symbiosis of different technologies to get more out of from the single cell.
Definitely, it's super interesting, the new technology. So moving a little bit, so we don't spend too much time here. I'm just wondering about maybe new techniques or new applications that are coming up in the field of flow cytometry quite recently. Any news to share, Christiane? Anything new, techniques or applications that you've seen so far?
So for me and for us in our facility, they may not be entirely super new, but we are getting down to trying out more nuclei sorting and doing RNA from nuclei. And I think one of the bigger things, especially that flow cytometry and omics, that we talk about, is in getting things onto the single cell level. And it's been out for a little while, but getting that resolution and combining that flow cytometry with your omics and being able to get pull that data and information from single cells. And so we talked about, spectral and then hybrid, and then now also more of that, okay, we're going to sort and then we're going to take it down to and do, like you said, 10x and moving forward from that as well. And so that's what I've seen a little bit more of. And Raif, I know from your experience, you've got a lot of that, the immunology and industry background as well. I'd love to also know what things that you're seeing too.
Yeah, absolutely. I mean, I agree with you absolutely with the single cell genomics and the single cell transcriptomics, and it's people want more information on the single cell level, and it's very important not only on the protein level but also in the transcriptomics level, what's happening on the single cell. So from the cytometry perspective, it's more about the cross facility collaborations with the genomics colleagues and the sequencing colleagues and our facilities to sort single cells. I mean, imagine we sorted a single archaea or single bacteria and then doing genomics on this single bacteria, single archaea, single fungal, single algae. That's quite powerful. And I think it's on the micro level, the flow cytometry on the micro level, like exosomes, endosomes, and the microparticles, or virometry, that's getting more challenging and exciting, actually. On the other side, of course, people on the immunological, the traditional application, adding more antibodies, adding more dyes, and getting more into the subset using low volume of samples. This is, I mean, the advantage of having the multicolor opportunities, 60 color or 50 color applications allows you don't need anymore so many samples. That means a small amount of samples will allow you to multiplex your flow cytometry. And I think from the technology, we do quite techniques. The future, I mean, we have the traditional flow cytometry application field, the immunology, but if we consider what's happening in the world right now, the climate change, the climate effect, and we want to get quantitative, or we want to track what's happening in the sea, in the oceans, in the earth, I think on the cellular level, on the particle level, that's where we get really more, these fields will use the unique features of the cytometry, the power to quantifying in a short time, multiple samples. And I think this is how we, and in this direction we will go in the next futures, I believe so. New fields and new challenges, actually. It's nice. It's nice challenges there.
Yeah, definitely. It looks like, Christiane, you do a lot of this, right? You don't really do much immunology, but you do more work with smaller single cell organisms, right? Right.
Yeah, we do. So we have at our campus in Bloomington, we do have immunological samples, but there are fewer. We deal, like you were saying before in the introduction, that we deal with archaea and microbes, C. elegans. So there we've done sorting for RNA extraction for C. elegans neuronal cells, and looking at for example, Arabidopsis plant nuclei, and looking and doing sequencing. And you mentioned, so my background is actually in Drosophila and looking at endoreduplication. So when you have that cell cycle and you get 2C content, double 4C, 8, 32 without the cell ever dividing. And we have groups who work with that to see it and because that's common in cancer cells and in developmental stages. And so we definitely work with that. And Raif, as you were talking about with the environmental samples, and there are flow cytometers right now on boats, and they are sampling and they're moving across. And like you were mentioning, what's happening right now in the oceans and other bodies of water, they're able to sample that. An advantage of that is for most of the time, they don't need dyes. They have naturally occurring fluorophores so they fluoresce inherently, and that's pretty amazing too. Just because they can take a sampling, and based on light scatter and based on inherent fluorescence in the cells, they can then parse out, okay, what populations do we have in there and how are they changing over time? And so these are longitudinal studies. They're over a long period of time, and again, cytometers on ships or cytometers that are monitoring in the ocean and in bodies of water. That's pretty fascinating.
I mean, it's so awesome to see when a researcher is coming from a non-traditional flow cytometry field, from a marine or from environmental, or we have colleagues here doing the Galeria studies, and showing them what is the capability and then the potential of flow cytometry. There, you see the eyes, and then you said, that's it. That's why I love flow cytometry. That's why I'm in this field. And I fully agree. I mean, this is awesome. The point is that at the moment, it's still the majority of the funding is going towards the pharma companies and towards the human medical research. Yeah, but I'm pretty much sure not very long time, and we will see there is also a big direction going fundings into the climate change and environmental research. So people will push a lot of developments in this direction for flow cytometry as well. So it's great.
Yeah,
I'm jealous that you can start C. elegans, and I'm still. I tried this in Aberdeen. Not really worked out, but I'm hoping to get one of these macro sorters as well. So we have we have here a really massive fly labs and Drosophila research going on here, and that will be probably. And then I'm coming to you.
Yeah, I think some things someone told me, and it was, and I don't remember, it was somebody who was coming through our facility. I was giving them a tour and I said, we'll put anything within reason through our instrumentation as long as it fits and as long as it's not above biosafety level 2. And the person said, oh, so if it fits, it flows. And I said, yeah, I think that was perfect. And again, I don't know who to credit that to, but if it fits, it flows. And
I
so
think you should take it and just trademark it.
Trademark it, yeah. Because we do, I mean, we have immunophenotyping, stem cell research, but you have microbiological research, looking at genes. We have groups here on campus that do ovarian cancer research, so we're doing a lot of stem cell sorting, but there's so many uses for flow cytometry. Like you were saying, Raif, there's more than just immunophenotyping. There's so much more that we can expand out to, and that whole if it fits, it flows. We'll try it. And we'll work with it. We have a group who works with snails that they bring over from New Zealand. So yeah.
Fascinated. We did this last week. A snail. I was really fascinated from colleagues from the Penryn campus here in Cornwall. He brought live snails. Just let me explain this really, Sam, that's really, he brought the live snails and then he poked underneath in the snail. And then they are secreting a solution, a foam. That's a defense mechanism. It smells. And then it's also, you need to try that. It's a yellow-green. Just solve this in PBS. It's getting high viscous and you need to dilute this. And there are specific bacteria. It's a symbiosis of the snail and the bacteria that he's discovering and wants to quantify this bacteria, analyzing this bacteria. It's awesome. It's fascinating. Yeah. I mean, at the end of the day, this is physical instrumentation. Who is telling that it's only limited into the immunological world?
Exactly. Exactly. Anything that can scatter light can go through it and you can analyze it, right?
Yeah,
absolutely. Absolutely. Yeah. Just to change gears a little bit, instead of research, could we talk about lab management? You are both core lab managers, so we might have a few present on the attendees, so maybe they're curious about how you're running things. So are you willing to share any tips and tricks or best practices when handling these sensitive cytometers? Could you start, Raif? What can you share?
That's the beauty in the Cytometry Society. Nothing is hidden, everything is public, everything is shareable, and everything available. So you will find a lot of information, this kind of information, if someone wants to set up a facility or manage a facility, you will find a lot on the International Society's website, ISAC. There's a lot of information over there. There's a lot of committees about shared resource lab, about data management, about all this. But absolutely, yeah. I mean, I can tell you from because we set it up. I set it up this together with colleagues from the technical service. I mean, they were really amazingly helpful here at the University of Exeter, setting up this new Cytomics lab and cytometry lab. So it's very important before you are setting up a lab, when you set up a new lab, it's very important to know what kind of research is going on around the application research field around. First of all, I don't want an instrument that cannot be used. Not just to tell, oh yeah, it's a fancy machine, let's have it, and then yeah, nobody's using it. Yeah. No, it was very important that we have instruments that so selecting, and of course you need to have the labs also in a specific structure as well. You need to consider. I'm always a big fan of dividing analyzers from the sorters, because in case of sterility, in case of you are sorting some sterile species. And there is very important that you are really from the beginning considering what kind of applications, what kind of QC protocols, what kind of SOPs, standard operating procedures, regulations. So these are things that people need to consider. The first thing what I did as well, because we are a self-recovering facility, it's also very important, the booking, scheduling, managing, financial rules and setups, because a facility is not managed just on a paper that was maybe 200 or 100 years ago. Meanwhile, there is much easier going to online digital systems. So the first thing what I have done was contacting Tobias from Cluster Market. And I had iLab system in Aberdeen, and there is also other managing system. But the advantage here was I knew Tobias and it was free, and he immediately set it up. So the booking, scheduling, invoicing system, and we were up to run. So from one day to the next day, we were ready to go from the managing perspective. And I was a one-man show at that time when I arrived here. So it's very important that you are really considering this. So it's very important to chat with the expertise at the university, finance people, technical service department, purchasing departments, and also get in touch with the researchers, with the PIs, speak to them and what is necessary. And yeah, that's from the beginning what I would recommend people to do. I'm sure Christiane will tell things that is probably in common. Please go ahead, Christiane.
Yeah, so I totally agree with space-wise. So it's super important. If you have any room, especially if you're first designing a facility. I was in a situation where we actually had just a caliber to start out with. We ended up getting 2 instruments added, an LSR2 and an ARIA, because we had a couple of researchers coming in who wanted and needed those instruments. Because as much as a caliber is great, and ours has since been retired, and ours did sort once upon a time. If you've ever had a caliber that sorted, then I feel for you. But it did sort. But Raif, as you were saying, it's the needs of the researchers. And actually our flow facility came about because of the needs of incoming researchers. And it expanded to help with the needs of any of the researchers that were coming, whether they were in psychology, kinesiology, physics, biology. So our facility is located in biology. We have chemistry, biochemistry, optometry who come and use the facility. And so, yeah, I totally agree with you, Raif. That's super important, is to know your researchers and what their needs are. We've since expanded. We have a Miltenny Max Quant vibe. It's actually on a cart. So maybe that's one consideration of mobility. If you need to, if you have an instrument that can be that's smaller, that you can move around, and we use that for teaching as well, as people will do long-term. If they need to take it for a day because they have time courses, they'll use that. And we also have an SH800. It's a self-service sorter, so people can learn how to use that, as well as the Copas Select system that we inherited from a Drosophila lab. But yeah, space-wise, initially they were going to put us into a space next to an autoclave room, which would have been a very bad idea. And we would have had air conditioners hanging from the ceiling and potentially, catching water and dripping. Thankfully, a space opened up in the basement, but something to consider is ceiling heights. Our facility is 7.5-foot ceilings. We cannot put biosafety cabinets. We have to do biobubbles. And if we need to, because clearance for a biosafety cabinet for some of the instruments is we don't get the clearance, and we can't hook it into the HVAC system because of how the ceiling is. And so we have instrument in a biobubble. Oh, wow. So that's something to keep in mind. Also, heating and cooling. We have an auxiliary unit in another room that pumps in so that our facility does not get above 75 degrees Fahrenheit in the summertime, and because otherwise it could be 80 degrees Fahrenheit in the facility. I don't know exactly what that translates into in Celsius, but warm and not comfortable for either researchers or the instrumentation. So yeah, and that, like you said, that space consideration, we are lucky. I'm lucky in my lab. I'm the only person in the lab, and so I'm currently a core of one staff member. That is myself. But we're lucky to have that space for all of the instrumentation and good facilities. Get to know your facilities, people. And become friends with them, so that when something does go wrong, then they can come and help out. And like you said, scheduling, tips and tricks too. That's facility setup wise. Of course, we can go into, make sure you filter your cells. Don't give us snot, please. We're not going to put that through the instrument.
What Shapiro saying?
Yeah.
Punkt out.
Yes, garbage in, garbage out. Mm-hmm. Yeah. If you have not seen the book Practical Flow Cytometry by Howard Shapiro, Raif, you're gonna pull one off your shelf. Yeah.
I have the second edition.
Oh, I think mine's over in the facility. So,
all right.
Yeah.
Look, this is the second edition from Howard Shapiro. Unbelievable. And look here, it's handwritten. Oh, damn! I can.
Yeah,
a little bit.
That's the Bible.
Yeah. Yeah, and that is available online for free as well. So if you look up, if you go to, I think Beckman Coulter houses that, and then that's an amazing resource. There is, have that. You don't have to read it front to back, but it's there and it's available, and you can find Howard's rules of flow cytometry, which, as Raif pointed out, garbage in, garbage out. And one of, I think, the most important things, and the IU Bloomington Flow Cytometry Core Facility has a Twitter account. It's talk to your core manager and talk to your core staff. We are here for you. We are here for you, and we want you to succeed. If you're a researcher who's using a core facility, so that in terms of those who are coming to the facility, or if you have your own instrument, feel free to reach out to core managers and other people in the field, because, and I know, Raif, you'll agree with me, flow cytometrists, we are a very open, welcoming community. And as Raif already mentioned, there's so much information already out there. Yeah. So utilize your resources and come see us.
Sounds good. Thank you for those insights. I have a quick follow-up question to this. So if you were to set up a new lab today, both of you, what would you do different? What would you do the same that was a success in your cases? Would you start, Raif, sharing?
Probably I would start in a similar way, and but I would consider room from the beginning, more room, because I should have from the beginning, although we have 2 rooms right now and also a computing room, but I realize we are getting an issue if we consider that we are still in the process of getting new instrumentations. So that's what I would recommend, or that's really, once you set it up and then you're realizing you are limited on the space, it's going to be difficult to find space. Universities in general, I do understand there's a lot of research going on, a lot of info and changes are going on. But so it's very important that you are from the beginning also starting to have the proper space and proper room to set up your instrumentations. And yeah, it's also depends on how you are starting a new lab. Is it something that you are starting from a scratch and then maybe giving you the opportunity, the financial aspect is also very important, because you don't want to deal with any financial issues from the beginning, writing a grant or writing any funding to any funding bodies. You want to set up your lab. So it's important whether there is a capital investment projects and capital investment opportunities in the university as well. And also, yeah, if there's a chance you are able to negotiate as much as possible. You can
so this is with the companies, right?
Yeah, with the companies, but also for the funding bodies or the university. Negotiate as much as possible, because once you signed, everything is over. It's difficult then to go back. Of course, the universities. I'm really here at the University of Exeter. I'm quite in a comfortable condition a bit. And they have a scheme of capital investment as well, which you need to apply. Of course, you need to apply for it, and that's not bad. And yeah, that's what I would recommend from the beginning.
Sounds good. Thank you. How about you, Christiane?
Yeah, again, I agree. Space is important, and the room to grow, because especially in a university situation, you're with new faculty coming in, and space can be restricted. So I agree with get as much space as you can in the beginning, especially if you're going to be adding on to instrumentation, with instrumentation. And also, if you're dealing with an existing space and making modifications, making sure that you have outlets where you need. Yeah. And making sure, like I said, in our case, I would move us out of the basement into a place with a higher ceiling that would fit us. We have a lot of space, but if I tried to fit a couple more instruments in there, then we'd start getting tight. Because I have our space that we've been in for quite some time now was originally designed so that there are no benches in the middle of the room, which makes it helpful for mobility, but doesn't make it helpful for electrical. We've had to have electrical drop down from the ceiling. And there were no benches around the room, so we could put in tables. And the nice thing about that was that makes field service happy,
Mhm.
because they can access all the points of the instrument that they need to. Whereas if you had an instrument that was back to back, so then that's a little bit more difficult. So designing, like I said, designing that space and making sure you've got the funding in place, heat loads. You can stick 5 or 6 pieces of equipment into a smaller space, but then again, it gets hot. You have to, so think about the cooling, and think about what your buildings, and talk to, again, your facilities people, and see what your buildings can handle and how the cooling works in your buildings. If we had a day where the chill water, so how it works, the university uses chilled water to cool the buildings. And so the chilled water went down for the entire campus. We were lucky because we had an auxiliary air conditioning unit that pumped in cold air. And so we were comfortable and the facility was comfortable. And the reason why the reason for the heat load, because I keep talking about this a little bit, because you have a lot of metal in your equipment. So that's one thing, is that if your laser is sitting on a metal mount, what happens if it expands and then it contracts? Slight movements, and so you're no longer in alignment, potentially. I've seen it happen when it's gotten hot in our core facility. And other things, the equipment doesn't like the humidity very much either, so you might have to get a dehumidifier in the summertime. You might have to get a humidifier in the wintertime. And so things like that, is that even if you're designing a new facility, there might be limitations depending on the instrument, that the building itself, and having HEPA filtration, things like that is helpful as well. And like you said, having those scheduling tools in place, and because our university also has iLabs, which I have not gone to iLabs. So I mean, it is it's a very versatile tool. We haven't personally used it, but that's, again, scheduling systems, accounting, talk to your financial people, and again, that's on the side of setting up the core. And there's other things to consider as well, as know your accounting person, know your purchasing person, and also having relationships with consumables reps, having relationships with not only instrument reps and your instrument people, but consumables, like, that sheath fluid, probably want to order that in advance, and if there's a shortage, might have to adjust your time frame depending on how much you need. And if our department also has media prep lab. Maybe you're going to have to get some PBS from them. But again, build those relationships. Those are important.
Yeah, absolutely. We have a quick comment from someone from the public saying that an external source of air pressure is a treasure for flow labs.
Oh yeah. I mean, it's something that should be considered, because if you have a pressure or compressor in the lab, it's noisy, crazy noisy. So we are considering. We have one instrument that has a compressor in the sorter room. So we are considering to put this compressor outside, because the air pressure, that's another factor. We have an internal air system, we have it, but the pressure is not sufficient. So that needs to be considered. Plus the air quality. So the air quality and the air pressure is not sufficient, because you don't want to get anything into your instrumentations, any particles. So that's I would recommend as well this considering to put the compressor either outside, or if you have an opportunity to install an internal air pressure system with a clean air, that's very valuable.
Yeah, absolutely.
And one thing that I would really, I mean, I'm in a good position. I was in a good position to get also some team colleagues. That's something, if you have this opportunity, select the right people, get the right people on board. And I can tell here my story. I wanted one particular person because I used to work with him in the past. At that time, I was angry to him. He left and he cheated on me. His name is Attila. You met him, Sam, Attila,
Yes,
baby.
I did. Yes.
So we used to work in Aberdeen together, and then he went to Rachel, into Babraham's Institute, to Rachel Walker, and then yeah. So when I moved down to Exeter, I said, Attila, I want you back. Come back, man. So no, I'm really glad to have a team colleague that is really, the chemistry fits, and he's a cytometrist, in his little bones, in the little cells.
Mm-hmm.
And nowadays, it's not only more the technologies that you want, it's more also about the computing science data. We had, for example, an internship right now, a mathematician. It's sometimes really very important because, and at the moment, it's very valuable because it's all about, meanwhile, the data science, which needs to be modified and to develop. I think that all this needs to be considered also when you're setting up. Plus, network. Get in touch with Isaac.
Absolutely.
Get in touch with Cytomics. Get in touch. I mean, you mentioned this as well, Christiane. I mean, the local. You are in the local associated societies and with the international societies. So they are helping, they are supporting, they are guiding, and they are next to you.
So going off of this, what you just said, Raif, I went to, Raif and I, we both attended Flow Cytometry UK last week at Birmingham University. Beautiful place, by the way, if you ever get to go there. But I was amazed. It was my first time in the event, and I was amazed at how close-knit the community was and how everyone knew each other and they used to work each other. Apparently people steal each other's, staff, but other than that, it was very friendly, and they were also welcoming to the newer technicians and the newer core managers that were there for the first time as well. And even to me, which I'm from industry, I'm not a cytometrist. I have a biochem background, but I've never worked with a flow cytometry machine. And it was wonderful, truly. So for those people that are out here listening, would you mind sharing some of the local communities they could join both in the US and in the UK and Europe, please? Maybe we can start with Christiane.
Yeah, absolutely. So it's already been mentioned before, but ISAC. So ISAC is a big international cytometry community, and that has a big meeting every year, but also has the Cyto University, where you're going to have webinars and different seminars and get to learn more about that, about cytometry as well. And then they're affiliated societies across the world. So ISAC has affiliated societies across the world. Locally, so I'm in Indiana in the United States, and we have the Great Lakes International Imaging and Flow Cytometry Association, which I'll put in a little plug if you're in the area, that we have a meeting September 16th through 18th. Full disclosure, I'm on the board of directors for that. And
please
so
attend.
Yeah. Yeah. And just, again, various regional meetings within the United States, again, across the world. There are affiliated societies in various countries on different continents. And so, like I said, with ours, I like to plug, of course, the regional one because I'm in the area. But then also not only those societies, but get on the Purdue Flow Cytometry listserv. That is such a wealth of knowledge that's on there, and you'll get so many people answering you and the experts are there. And they take the time to answer. It's amazing. When I was first starting out, I had a plant question. I'm like, why can't I set up cell cycle on Arabidopsis as well as I can with a mammalian cell? And David Galbraith answered me back and said, I'm going to put you in touch with somebody in my lab who's going to help you. And I was like, oh, yes, thank you so much. And, that was my first foray into that. And it's like Sam, you were pointing out, it's a very welcoming and open community. And so don't be afraid to ask questions, and join join a society or ISAC, both, all of them. Another that I would recommend for core managers and core staff would be ABRF, the Association for Biomolecular Resource Facilities.
Mhm.
And so that includes flow cytometry, but it also includes microscopy, genomics, and more affiliations there. So that's a great way to talk to other people who might be using the cells you sorted.
Yes.
Along the way too. And there's an international community there as well.
Okay, well, how about you, Raif? Could you share something local in the UK and in Europe?
Yeah, actually, yes. I just wanted to put something on a chat. If you go on a isaac.net.org to the Isaac website, what I would recommend to everyone to get onto the Isaac website. On the Isaac website, you will find under the committees and task force, also a task force and subcommittee, meanwhile, the associate societies. And there you will find a lot of associate societies name. So associate societies are local societies from all over the world. So UK, Germany, in Germany is the German Society for Cytometry, Deutsche Gesellschaft für Cytometrie, für my German colleagues, the DGFZ, or in Turkey is the Turkish Society for Immunology. The cytometry is part of the immunology group over there. So, but you will find all this information, including the contact details for the Associate Society on the Isaac website. If you go there under the committee or actually associated websites, you will find resource, and there you will find the information. To get more about the cytometry in general, if you are in the university and you have a core lab, the core colleague is really the first contact which I would recommend to go. And then they will forward or they will provide the information about societies. So there is one in Europe as well, the ESCA, European Society of Clinical Cell Analysis, is a clinical cytometry in Europe. And what else I know? The Irish ones, Cytometry Society Ireland, CSI, the Scottish Society of Cytomics. And yeah, we have a local as well within UK, like London Flow Club, Cambridge Flow Cytometry, Scottish Society, but the general one is the Flow Cytometry UK. And then French has, or Iberian Society, the Spanish speaking society. So I would recommend go on a website, Isaac, you will find all this information there.
So definitely go to Isaac and look for it, because there's tons of local and bigger regional communities, and please join them, of course.
And also the Cyto University information. We have our own university. It's called Cyto University. That's really, but very, very, very useful.
Absolutely useful. Lots of great resources. I guess I'll open up the floor since I have 5 minutes. Anyone else wants to ask a question to Christiane or Raif, now is a chance for you to ask, and we'll be able to answer them before we finish up here.
Yeah. And I will also mention quickly, so at least in the US, we also have the annual course in flow cytometry that happens yearly, and that's a week-long course for those who are wanting to learn more and get their hands on flow cytometry, as well as there are different, and one that I am most aware of, there's, for example, the expert cytometry, where you can also learn more, and they have a course and you can go through. They have online presence as well, where you can ask questions on, they have a Facebook group if you are a part of their membership, where you can also ask questions too. So those are some other resources that are out there as well.
It's now with Cheeky Scientist.
Cheeky Scientist, yes.
Yeah, it's good. It's in the organization Cheeky Scientist. The expert side, X-side from Tim Bushnell is with Isaac and Cheeky Scientist now. Since you mentioned, we forgot one important one in UK, actually, the very important one, the Royal Microscope Society.
Yes.
RMS.
RMS.
Oh my God. I hope Karen is not out there. She will kill me that I didn't mention that. RMS is really important one.
Yeah.
Absolutely. So I don't think we really have any questions from the attendees. So I think I will wrap it up here and thank both Raif and Christiane for participating today. It was a pleasure and it was great to hear from professionals on long-term experienced cytometrists from what you're doing and where it is going and how other people can join as well, right?
Well, thank you very much.
Thank you very much. Yeah, it was a great pleasure here to be together with Christiane and discussing and chatting. Actually, it's nice. Cytometers like that, and anytime. So
Yeah.
it was a great pleasure, Sam. Thank you so much for this.
Yeah, thank you for the opportunity and the invitation to do this. This was great, and I think too, if anybody wants to reach out offline, I'm happy to talk to people as well. My contact information can be, you can find me if you look up my name and IU Bloomington Flow Cytometry, it's gonna pop right up. So
yeah,
yeah.
You can also find them on LinkedIn as well, right? And then follow them there for updates and connecting the network. Absolutely. I would like to also thank everyone that attended here. I see some familiar names, some new ones as well from all around the world. So thank you very much, everyone. Hope you had a great informative session and hope to see you next time.
Thank you.
Thank you. Bye-bye.
Thank you very much.
Keep calm and just let it flow. Good to see you guys.
Thank you. Bye-bye.
Good to see you. Thank you.